Quickly finishing off where we kind of got cut off in the last one. Bacterial superantigens, like Staphylococcus, produce these things called enterotoxins. These superantigens bind to the MHC class 1 receptor of an antigen-presenting cell. So if you see the image on the right here, the cell on the bottom, the antigen-presenting cell, they bind to the MHC class 2 receptor, but when they present this antigen to a T cell, the superantigen binds to the T cell receptor outside of its antigen binding site. So you can imagine normally it's kind of odd how they've spelt it here. They've pronounced it T cell. I might have got this image from a different language, but the principle is still the exact same. The T cell at the top and the antigen presenting cell at the bottom, you can see they both have receptors and there's a little gap in between them. That's where the antigen would usually be, but the superantigen binds to T cell receptor outside of the antigen binding site. This means that the T cells are going to get activated and begin producing many cytokines like TNF alpha, TNF gamma, interleukin 17, and interleukin 16 that would deal with an infection, and they're going to start dividing. But the problem is the T cells that have been activated are not specific to the superantigen. The superantigen hasn't binded to them and activated them from within the antigen binding site of their T cell receptor. So this means we're going to have the production of a lot of T cells that are not antigen specific. And all these non-antigen specific T cells are going to produce a ton of these cytokines, and these cytokines are going to in turn activate cells of the innate immune system like macrophages, monocytes, and neutrophils, which in turn produce their own cytokines like interleukin 1 beta and platelet activating factor. And because so many T cells and phagocytes are being activated by the superantigen, more than our immune system can handle, we're going to have septic shock within the person who this is occurring in. Basically, because, like you can imagine, normally T cells would only be activated for a specific antigen that is capable of binding to the antigen binding site. So only a small number of T cells are going to get activated. Only the T cells with complementary antigen binding sites to the antigen will get activated. But in this case, the superantigen is binding outside of the antigen binding site, so it's basically going to activate every kind of T cell that it comes across, regardless of the shape of the antigen binding site on the T cell receptor. This is going to activate so many T cells, they're going to produce so many cytokines, which are in turn going to activate so many phagocytes, which in turn produce so many cytokines that the body is just in shock. And then this is how we get our septic shock occurring. So hopefully that kind of made sense. We're going to quickly go over the immune response to fungi now, and then we're going to cut it off here. This is just a continuation of the last video. And then in the next one, we're going to talk about immunodeficiency diseases, and we're going to use HIV as the main example that we're going to cover. So, with regards to the immune response to fungi, ignore the picture I've put here, I just put it here because it kind of gets the message across. Fly agaric isn't a human pathogen, neither is Ophiocordyceps, shown at the bottom. I just thought they kind of get the idea of fungi and the potential pathogenic consequences of fungal infections, but they're not human pathogens. So the type of myeloid cells that respond to fungi depend on the pathogen. Remember, myeloid cells are just the broad cell group of the innate immune cells, like the phagocytes, of being the macrophages, neutrophils, monocytes. These myeloid cells get activated by fungal glycans or glucans on the fungal cell wall. Immune cells respond by producing inflammatory cytokines and by phagocytizing fungal cells. The adaptive immune system utilizes cell-mediated inhibition to fight fungal infections, with Th17 cells producing interleukin-17, which recruits and activates neutrophils and monocytes, as well as making epithelial surfaces producing antimicrobial peptides. So remember, we talked about Th17 cells dealing with extracellular bacteria and dealing with extracellular pathogens in general. And when we talked about the cell type, specifically when we're talking about different kinds of helper T cell, we talked about how they deal with fungal infections. Well, this is how the Th17 cells are producing interleukin-17, which is recruiting our neutrophils and monocytes to phagocytize fungal pathogens in the area, as well as producing epithelial surfaces, as well as producing antimicrobial factors on epithelial surfaces that are killing any fungal pathogens and preventing infection by more fungal spores on these epithelial surfaces. So obviously the antimicrobial peptides are destroying any microbes that they're specific to that are landing on these mucosal surfaces. So antibodies produced by B cells will also prevent adherence of fungal pathogens and neutralize fungal toxins, as well as mediating antibody-dependent cytotoxicity for natural killer cells. So the antibody binds to a fungal pathogen, which marks it for death. When a natural killer cell arrives, it binds to the Fc region of the antibody, and then this begins this antibody-dependent cytotoxicity, where the natural killer cell is going to release cell death signals and release granulozymes and perforins to destroy whatever cell the antibody has latched itself onto. Fungi can also evade the immune system, like we talked about bacteria and viruses evading the immune system, and they do this by secreting immunosuppressive cytokines that downregulate pathogen recognition receptors. So there are less receptors on antigen-presenting cells that are presenting these fungal antigens that are going to trigger an immune response. And fungal pathogens can also produce an anti-inflammatory cytokines that prevent an inflammatory response, as well as preventing phagosome fusion within the lysosome. If I talked a bit fast there, so it's kind of a dense topic to cover, I'd recommend just reading over the lecture slides. The next one, we're moving topic now from the immune responses to the different kinds of pathogens like virus, bacteria and fungi. The new topic is going to be on immunodeficiency, examples of immunodeficiency, and then we're going to talk about the opposite of immunodeficiency, being hypersensitivity reactions, where immunodeficiency, you're not getting enough of an immune response. Hypersensitivity reactions, you're getting an over-aggressive immune response. So we'll get onto that in the next few videos of the playlist.